Hello and welcome to Taconic's webinar entitled Cryopreservation, an Investment in Genetic Stability, Genetically Engineered Mouse Model Preservation and Cost Savings, presented by Dr. Philip Damiani. Please allow me to introduce myself. My name is Margit Wissenbach and I will be your moderator today. Before we get started, I'd like to address a few practical matters. Today's webinar will be made available to all attendees. We will email a link to the presentation to you by the end of this week. Directly after the presentation, Dr. Damiani will answer your questions as they appear in the control panel. So if you have a question, please type it in the control panel anytime. The other attendees will not be able to see your question as it is sent directly to Dr. Damiani and me. If you cannot see the control panel, just single-click on the single arrow which is located on the top of your screen. If your question is not answered at the end of today's session, Dr. Damiani will respond to you within the next 24 hours via email. In case you're experiencing technical difficulties, please raise your hand in the control panel and our staff will assist you. So, let's get started. We hope you will enjoy today's webinar, Cryopreservation, an investment in genetic stability, gem preservation, and cost savings, presented by Dr. Philip Damiani. Dr. Damiani joined Taconic in 2006 as senior embryologist. He is currently head of global embryology and is responsible for coordinating scientific research and development within Taconic's embryology laboratories. Dr. Damiani holds a doctorate in reproductive physiology from the University of Massachusetts. Prior to joining Taconic, he worked extensively in the field of assisted reproductive technology, including sperm and embryo cryopreservation, in vitro fertilization, and nuclear transfer for a wide variety of species, including domestic and non-domestic species. He spent some time in South Africa working with other researchers to establish a biobank and an assisted reproductive laboratory for endangered species. During his career, he has assisted many zoological parks and laboratory animal facilities in developing disaster plans for ensuring the safety of both living and cryopreserved materials. In this webinar, Dr. Damiani will review the basic concepts of embryo and sperm cryopreservation of rodent models. He will elaborate on some of the advantages of cryopreservation of your model and how this plays a critical role for colony management. With this, I will hand over the word to Dr. Damiani. Please, Dr. Damiani.
Good morning. Today we were going to talk about cryopreservation, an investment in genetic stability, and gem preservation and cost savings. I just want to give a little bit of background about Taconic's assisted reproductive program so that you can kind of get a feeling as to what we're capable of doing. We actually have an embryology laboratory set up in our main facility in Germantown, New York. We also have a duplicate embryology laboratory in our Denmark facility. It is staffed by 2 full-time laboratory managers and 1 full-time embryologist. In the laboratories, we're capable of incorporating multiple assisted reproductive techniques, including in vitro fertilization and embryo transfer. And we also have a full functioning cryopreservation program, and that's particularly about embryo cryopreservation, sperm cryopreservation, biobanking, or what other people know as repository storage. And that's today what we're going to talk about, particularly our cryopreservation program. What I hope to do in this webinar is to give you a better understanding as to what cryopreservation is, why you would want to cryopreserve your samples, what are some of the advantages of using cryopreservations, what are some of the recent advances in cryobiology, and then to summarize what is a biobank and a repository. So cryopreservation, it's basically the science that permits low temperature maintenance of a diversity of cells, and these cells can be anywhere between gametes such as sperm and eggs, or they can be somatic cells such as fibroblasts or embryonic stem cells. The main objective of cryopreservation is that you want to be able to minimize damage of the material during the low freezing process and storage. And this is very critical because this will really determine your success rates and the ability to rederive the material after it's cryopreserved. There are many different types of cryoprotectant agents, or what we call as CPAs. We're not going to focus today on all the multiple cryoprotectants that are out there, but the main point to take home is that they basically protect the cells by reducing cell shrinkage, and they also reduce the fraction of solution frozen. So what this really means is that it minimizes intracellular ice formation. And the way that I think that most people understand this is, for example, when you make ice cubes and you place them in an ice tray, the liquid water basically is filled in the ice tray. But after you freeze it for a period of time, the water molecules line up, and then the actual ice cube has a tendency to expand and pop out of the ice tray. And that's what's happening in cells when you basically are cryopreserving them. If you don't add a cryoprotectant to help minimize that expansion of the water molecules, you wind up with the cells becoming damaged, or what we call lysis, and then your material is no longer viable. So the cryoprotectant helps prevent that, or I should say minimize that intracellular ice formation. There are 3 known methods for cryopreservations. One of them is called the control rate or slow freezing method. That's basically where you have sample dehydration and you have ice crystal formation occurring. Another method, and I should go back, controlled rate freezing is commonly used many times with embryo cryopreservation. So you basically have a decrease in temperature, a set amount of temperature decreasing over a set period of time. So for many embryo cryopreservation protocols, it's usually a ½ degree per minute until you get to a particular temperature, and then you plunge those embryos into liquid nitrogen. That plunging process is known as rapid or non-control rate freezing, and that's where you get a large drop in temperature over a very quick period of time. Rapid or non-control rate freezing is typically also used in sperm cryopreservation, in which you are floating your samples over liquid nitrogen vapor phase for a set period of time. Again, the temperature drops very rapidly over a short period of time, and then you follow it up with a plunge into liquid nitrogen. Another method that's commonly used in the industry is what's known as vitrification. And vitrification differs from both controlled rate freezing and rapid freezing in that there's no ice crystal formation occurring. You basically get what we call the material being frozen in a glass state. So it's basically very clear. The cryoprotectant, when it's frozen, is very clear, and that's the reason why we call it the glass state. One of the disadvantages of using vitrification compared to control rate freezing or rapid freezing is the concentration of cryoprotectant needed to achieve that glass or non-ice crystal formation state. The cryoprotectant used in vitrification, the concentrations are extremely high, which means that if you don't work very quickly with your samples, you have a tendency to cause toxicity within your biological specimen. So for example, with embryos, when you try to vitrify your embryos, you need to be able to work very quickly in moving your embryos through the different cryoprotectant agents. Otherwise, at 37 degrees centigrade, they have a tendency to metabolize, the embryos have a tendency to metabolize those cryoprotectants and eventually cause some toxicity and then decreases your viability post-thawing. So there are multiple methods for cryopreservation. So why would you want to cryopreserve some of your rodent colonies? And the main reason is that disasters happen. As we all know, our colonies are pretty important to our researchers and our PIs, but they're also very prone to many disasters that are out of our control. There's obviously natural disasters, such as floods and hurricanes, fires and earthquakes and tornadoes, depending on what part of the country that you live in. But there's also many man-made disasters that we're not aware of. Those fall under animal husbandry errors, such as genetic drift in an existing colony. So your animal care technicians may not be paying attention to a particular line, or there may be some mismatings and you start getting a genetic drift. You can also lose phenotypes over a period of time. Again, that's considered a genetic drift. There's also genetic contaminations where you would have mismatings or improper setups. There's also animal rights activists. They can be an issue when they're particularly dealing with man-made disasters. And also there's facility issues such as power outages due to natural disasters such as ice storms or floods and hurricanes. So one of the things that we would like to emphasize, particularly not only when you're dealing with a cryopreservation program, but is also to consider disaster planning. And these are just some of the steps when we're talking about setting up a disaster planning or a disaster program. So you need to be prepared, you need to mitigate, you also need to be able to have a response, which is the actual activity of the emergency plan when the need arises. And then there's also the recovery phase, and those are the actions needed to restore normal operations. And it's where in the recovery phase of a disaster planning where cryopreservation plays a critical role. There's also other issues with animal husbandry in which there are many variables associated with running a colony. And I believe that cryopreserving your samples will help with some of these animal husbandry issues. So one of the issues that occurs, obviously, is that expansion of facilities. Space and budget restrictions prevent you from expanding your colony, so you basically run out of space. So being able to remove existing colonies, cryopreserve them in a bank, and then rederive them as needed should help with the expansion issue or lack of space. It's also very difficult to hire and retain employees, so you may notice that you could have a lot of human-made errors as staff gets rotated through your colony. So again, having cryopreserved material as a backup will help in the event you have genetic contamination or genetic drift due to employee issues. There's also the veterinarian and animal health issues. As everybody knows, it's very difficult to bring in new live animals into a colony, particularly because of the risk of pathogens associated with those animals. And it also makes it very difficult to share material among other collaborators. But being able to ship cryopreserved material, that will also be a lot easier, and we'll explain some of that a little bit later on in the webinar, where you don't have this risk of contamination and you don't need to quarantine animals because of the cryopreserved material. We also know that animal husbandry is extremely expensive, and then this is just an example that we put together in regards to a maintenance colony at just a flat rate of $1 a cage, having 5 cages as a maintenance colony. Over a 3.5-year period, the cost of that line is about $6,400. If you cryopreserve that line within the first year or so of maintaining it at the maintenance level, you would save about almost $3,000 in cage costs. If you went forward with sperm cryo, you would save about $4,600 in cage costs. And then you would be able to just go back when you needed to and rederive the line in the event that a PI or an investigator wanted to do some additional studies. It's also very expensive to generate many of these genetically engineered models. For example, it's about $100,000 to generate a line using ES cells to generate the chimeric animals. And this is obviously the total cost, including the manipulation of the ES cells, the culture, and so forth. There's also very long timelines and many variables associated with the generation of these genetically engineered models. Having some type of material cryobanked, whether it's the ES cells that are already manipulated or the actual sperm from some of these animals, could help alleviate cost in the event that you need to restart the project over again. So we're going to talk a little bit about some of the applications of cryopreservation and which types of methods that are currently available. So some of the advantages of the cryopreservation is that when you cryopreserve gametes and embryos, you can't prevent the genetic drift in your colony because you basically are taking a snapshot of that colony. It offers an alternative method into maintaining active breeding of your colony. So instead of having your maintenance colony or your maintenance cages, you can cryopreserve the stock and pull it out of the repository as needed. Another interesting part of cryopreservation is that it increases the generation time and allows further contributions of your model's genetics way past its reproductive lifespan. So for example, you're able to cryopreserve material and keep it stored for 10, 15, 20 years down the line, a lot longer than the natural reproductive lifespan of a mouse. It also allows for ease of transportation of the genetic stock. Like I just explained, it's a lot easier to ship cryopreserved material than live animals because there's no need for quarantine. It also decreases the risk of disease transmission, and more importantly, it allows for speedy recovery of stocks following any type of disasters. So some of the applications of cryopreservation, obviously there's embryo cryopreservation, and that's the most commonly used method. The most important thing about embryo cryopreservation is that zygosity of the line is preserved. So for example, if you have a homozygous knockout, you can produce homozygous embryos and then rederive those lines after it's been cryopreserved and have homozygous offspring immediately after thawing out your samples. We offer two types of cryopreservation at Taconic. It really depends on where you're doing your cryopreservation. We call ours homozygous embryo cryopreservation and heterozygous embryo cryopreservation. The difference between those are just basically the genetic background of the donor females. So if you're using wild-type animals and you use either a heterozygous or a homozygous male, we label those as heterozygous embryo because obviously you would not have homozygous offspring produced until you do additional breeding. There's also sperm cryopreservation. This is becoming extremely popular in the industry right now because it generally requires fewer animals needed. Typically 1 to 3 males would be required for sperm cryopreservation. It's also less expensive than embryo cryo, and you require less rebanking of the material. What that means is that when you cryopreserve semen, you have a lot more samples and a lot more straws than you would potentially if you cryopreserved embryos. You then thaw those straws of semen, produce additional embryos for your derivation, and those are what produce your offspring. So you don't necessarily have to go back and rebank many of the material because you're generating new embryos every time with the cryopreserved sperm. As opposed with embryos, you have a set number of embryos in your bank. The most important thing about sperm cryopreservation is that genetic matters when you're considering sperm cryopreservation. So for example, multiple versus single mutations may affect the ability of the sperm to cryopreserve properly. And you also require additional breeding upon recovery. Obviously, like I said, if you're trying to produce a homozygous line with sperm cryopreservation, you may be able to cryopreserve sperm from a homozygous male, but unless you have those donor females around, you're going to obviously have to use wild-type animals, and then those resulting offspring would be heterozygous, and additional breeding would be required to get back to the correct genotype. So we currently use a raffinose and skim protocol for our cryopreservation method, but we incorporate a beta-methyl cyclodextrin to the capacitation media. And this slide just basically is showing some of the improvements that we made in our sperm cryopreservation process, and that the fertilization rate from cryopreserved sperm now on multiple background strains is very similar to those that we obtain with fresh semen. So this is another reason why, as laboratories are getting very efficient in sperm cryopreservation, there's a change over from embryo to sperm cryopreservation. So the material that you would use for after sperm cryopreservation is generally, at Taconic, we offer the ability to do what we call a standard derivation with cryopreserved sperm. The straws contain usually 10 microliters of the cryopreserved sperm, and then we inseminate a certain number of oocytes to produce those embryos, and then do the transfers, and we will guarantee a minimum of 6 offspring following the use of sperm cryopreservation. There's also other material that can be cryopreserved in your system. One of those would be embryonic stem cells. Obviously, everybody knows that embryonic stem cells are used as vehicles to modify the genome, and they're capable of contributing to many different tissue types in the chimeras produced, including the germline. And that's what you want, is germline transmission. So you can cryopreserve your ES cells in the event of a disaster, and then be able to thaw out these cells and generate additional chimeric offspring. But another means of improving timeline and improving efficiency is that if you cryopreserve embryos such as morulas, which is the stage just prior to blastocyst formation, if you start banking many of those material, you can then start thawing out those morula, culturing them to produce the blastocyst, and then following up with injecting them with your ES cells. Again, minimizing the cost associated with having a colony necessary to superovulate for blastocyst formation, also minimizing timelines in regards to inefficiencies in the numbers of embryos collected following superovulation and natural mating. So it's just another means of shortening the timelines and generating more animals with less cost. There's also embryonic germ cell cryopreservation, and this is where you would cryopreserve the primordial germ cells. These can be either manipulated or non-manipulated. After thawing these samples out, they can be injected back into the testes of a male, which will allow propagations of the germ cells. And this has been very successful in reestablishing fertility in some lines in which low fertility or no fertility is observed. So you can take primordial germ cells from one particular animal and then inject them into another animal that has a higher fertility or a higher breeding efficiency, and then be able to generate those offspring. There's also the ability, I've done a lot of work with somatic cell nuclear transfer, so obviously you can cryopreserve your fibroblast skin cells. Again, this is just another method in which genetic modifications of the cells can be possible, and then those can be cryopreserved. And then once you thaw these cells out and generate those offspring, you wind up with your transgenic offspring. Again, minimizing timelines and costs associated with that. So what do you do with all these cryopreserved materials, whether it's embryos, sperm, ES cells, or somatic cells? You should consider establishing what we call a biobank. And biobanks are just typically a cryogenic storage unit, and it maintains these samples for a period of time. It allows just a method for collecting and storing and also distributing these samples. And these cryobanks can basically be the size of how large or how small you particularly want them. So there are commercially available biobanks, there are governmentally funded biobanks, which are large warehouse size facilities, or they can be single refrigerators or freezers, depending on what you want to establish. The most important about biobanking or repository storage is that you should send your samples to an established repository if you're choosing not to do it yourself. And what that allows you to do is allows dual site storage of your cryopreserved materials. So if you choose to keep some of your material in-house, you should then follow up with sending half of your material to some other site so that in the event of a disaster at your facility, you don't lose everything that you've cryopreserved. It also allows for easier exchange of material with other collaborators. So if you go and deposit your material into an established repository or an established biobank, generally those individuals that run that biobank will allow for the exchange of material and do all the upfront work, including the paperwork and all the permits needed to get the materials overseas. If you choose to establish your own biobank, you need to realize 4 important aspects. You need to understand the goals of your cryobank. What type of collections will it consist of? Is it only going to be embryos or it's only going to be sperm? You also need the support of the key stakeholders, whether that's the researchers in your facility, the community, the research community at your university, for example. You also need to consider funding. It needs to be a specific amount and a specific time frame that you have funding available, because you need to always replenish your materials with fresh liquid nitrogen. You need to establish the freezers and the database needed to maintain your samples. And you also need to have operations into consideration, whether that includes all the staffing, purchasing all the equipment, and then again, like I said, having a way of being able to record your specimens. So in conclusion, cryopreservation of lines allows for an effective means of collection, storage, and distribution of biological samples. Cryopreservation also plays a critical step in the preparation and the recovery phases of disaster planning. Like I mentioned, many different types of cryopreserved material can be banked, whether they're embryos, sperm, or cells, that will allow you to recover your genetic stock with minimum upfront cost. And banking of material requires both financial and collaborative support, whether it's internally or externally. And cryopreservation, or cryopreservation of your material, should be and really needs to be a part of your disaster planning program. So that concludes our webinar, and I'm more than happy to answer any questions that you may have.
Okay, thank you, Phil, for this very interesting presentation. So we will now start the questions and answers session. And we already have a couple of questions coming, but please don't hesitate to send more questions. We will be happy to answer them here or by email after this webinar. So the first question is, what type of quality control is conducted for cryopreserved materials?
Okay, there's multiple ways to do quality control for your cryopreserved material. So for embryos, we typically will cryopreserve a small subset of the embryos generated in its own separate straw, run them through the same protocol, thaw those embryos out, and either follow them up with doing an embryo transfer to generate live offspring, or typically we'll culture those to the blastocyst stage, and then again present that information back to the investigator. For sperm cryopreservation, we will thaw out a sample of sperm, do an in vitro fertilization, generate those embryos, and either culture to the blastocyst or again transfer those to generate live offspring. So it really depends on what resources you have available at your facility, but you should run some type of quality control to ensure that the material that you cryopreserved will be able to be re-derived at a later time.
Right. Another question is there any difference between cryopreserved material and non-cryopreserved material following embryo transfer? So any differences between those?
So if you have a very well-established cryopreservation system at your facility, you should not really be able to distinguish live born rates following the transfer of cryopreserved and thawed material versus non-cryopreserved material, there should really be no significant difference. You may notice that after thawing out your samples, for example, if you're using a stage of embryo such as a 4-cell or an 8-cell stage embryo, you may notice that you have some damage in a few blastomeres, but that should not really significantly affect your live-born rates, especially if you're doing everything properly and following up with quality control. Where you start seeing differences is where people have a tendency to rush through systems or may not actually have a properly functioning cryopreserved program.
Okay. Another question would be, I have a line that I have bred to homozygosity. Can I use sperm cryopreservation to preserve that line?
So yes, you can use sperm cryopreservation, but what you need to remember is that upon thawing the sperm themselves are homozygous, but unless you have the females available for the rederivation, you may have to follow up with using wild-type oocyte donors. And then again, all your offspring would be heterozygous and requires an additional round of breeding back to homozygosity. So if you're in a rush to rederive your line very quickly and don't want to wait around for an additional round of breeding, I would probably recommend embryo cryo for a homozygous line as opposed to sperm cryo.
Right. Another question is, of the 3 known methods of cryopreservation, controlled, rapid, and vitrification, which do you recommend or prefer?
So that's actually a very good question, because if I had to recommend a method or one that I would actually prefer, I believe in vitrification, particularly as a means to cryopreserve embryos. Now that said, it really depends on the type of program that you have. So for example, at Taconic, where we cryopreserve hundreds of embryos in a given day, vitrification is not really a method that we can incorporate in our daily operations, because like I mentioned before, the ability to be able to move samples very quickly through really prevents us to incorporate vitrification. If you're working in a smaller laboratory where you're only freezing maybe 50 to 100 maximum embryos, then again, I would recommend and prefer to use vitrification because it's a method that prevents ice crystal formation, which results in fewer damage to your material, and it has a higher success rate following post-thaw compared to control rate or rapid freezing.
Okay. Another question. So with regard to embryo, sperm, and DS cell cryopreservation, should all 3 be employed for a single line? If not, why not?
So if you have, I'm a strong believer in being able to cryopreserve or bank any biological specimen that you can get from your cell line, from your rodent line. So for example, if you've made these chimeric, you should be banking your ES cells. Upon the production of your animals, when you know you have germline transmission, you should go back and either cryopreserve embryos or both embryos and sperm from those founder animals. Again, because the more material that you have in your biobank, you have additional abilities to rederive the line. The other thing also to remember is technology changes over a period of time. So not many people thought, for example, in the cloning or the nuclear transfer field, that having cryopreserved material or skin cells in a bank would actually be able to allow you to regenerate a whole new individual. So I'm always a proponent of being able to cryopreserve back anything that you have in the event when you're tearing down a line. Even something as simple as serum or skin cells. Again, they could be useful later on to not only yourself, but some other potential collaborator.
Okay. Another question is, does Taconic offer biobanking storage?
Yes, Taconic does offer biobanking storage. So we would be considered a repository for whether a university or a pharmaceutical company. So what we do is we will take material over from a researcher and then also incorporate dual site storage in-house here. So we have a facility in our Germantown main facility where we cryopreserve the material, and then we also have dual site storage in our Albany, New York facility.
Okay. And another question, how long does it take to cryopreserve a line? How quickly can it be conducted?
So the time frame to cryopreserve a line depends on what type of method that you're using. So if you're using sperm cryopreservation, that will be cryopreserved in one replicate. So the males are euthanized, the material collected and cryopreserved. So again, that's within a 24-hour time frame. Obviously, then we do a quality control to ensure that it's passing, which would be done the following day. So within 48-hour time frame, you should be able to cryopreserve with sperm. With embryo cryopreservation, it really depends on what type of method you're using. We at Taconic will routinely use in vitro fertilization, so we try to cryopreserve 300 embryos in one replicate, and again, thawing out a sample to do our quality control and culturing it to a blastocyst. So the time frame would be basically a week from the time that the line arrived, cryopreserved, and having the results for our QC. So it really depends on what type of method you're using.
Okay. And maybe a last question here. Jackson actually offers IVF courses. Does Taconic do that too?
No, we have considered doing that. We have done some training in ALAS. Actually, just last year we did a cryopreservation training and a non-surgical embryo transfer. If people are interested in learning in vitro fertilization with cryopreserved sperm or just the process, that's something that we can consider and set up to do some training.
Okay, so I think that was our last question for the time being. Thank you everybody for your attention and for your questions. As mentioned in the beginning, we will send this presentation to you, and questions and answers will be on our webpage. And also, please keep an eye on further upcoming webinars. You can see those announced on our webpage, and they may also be emailed to you. So with this, I will say goodbye to you. Thank you, Phil, for the presentation. Thank you, everybody, for your attendance. Bye-bye.
Bye.